This video is on the sarcomere, um, the sliding filament theory of muscle contraction, and we'll point out the um, protein filaments that comprise the sarcomere. So in a previous video, we talked about how myofibrils run as these cylindrical units of contractile proteins along the lengths of skeletal muscle fibers. In other words, inside a skeletal muscle fiber, there are these multiple bundles, cylindrical in shape, that are essentially sarcomeres that are laid out end to end to end to end. The analogy I used was the cars in a train, essentially. And each one of these forms essentially the functional contractile unit of striated muscle. And so what we want to do is look at one of those units, because we want to understand how one works, then we understand how all of them work, and then all we have to do is sort of link them together lengthwise along a muscle fiber. And we can see how it would shorten end to end. While each individual sarcomere only shortens a tiny fraction, about a millionth of a meter, 1 micrometer. When you add up, thousands of them end to end, each one of them shortening by that little bit, the sum total of shortening can become quite significant. We can end up with, multiple centimeters of shortening in a fairly large muscle. So let's look at the arrangement of the sarcomere, and then we'll look at the protein fibers that it contains. And so the sarcomere is made up of lots of proteins, but the two main filaments, I should say, and this would repeat. I'm actually drawing only a portion of a sarcomere, because it repeats the arrangement here. I'll draw 3 of these thicker filaments, and then surrounding those thicker filament. The other thing is you have to think about this three-dimensionally. Like, we're looking at it from the side, and what we're gonna, I'll give you a sort of the idea if we were to cut through it and then turn it and look at the end of it, what the visual would look like. We have these other protein filaments that are thinner. Put them in blue. Short. And so we have these, so it's the theory of striated muscle shortening is called the sliding filament theory. So these structures I'm drawing here are protein filaments, right? So proteins that are polymers that form these long, sort of thread-like structures. And they're arranged and held in an array such that, kind of like your finger, if you took your fingers and did this, right, where they're sort of interdigitating. And so, and again, you can think of that as sort of three-dimensionally as well, right? Not just this two-dimensional version. And what we're going to find is that these filaments are going to pull past one another to shorten this whole thing. And so the idea behind, proteins are not rigid like your fingers are rigid, or pencils are rigid, right? You get pencils like overlapping. So because they're not rigid, we need to sort of hold them in space, and there have to be other proteins that will help them stay sort of aligned, right? And so they have to be sort of stabilized by an assortment of other proteins. And so there's this sort of stabilizing set of proteins at the that help arrange and hold the thin filaments so thin and thick filaments, right? Obviously, we'll give you more detail on those structures in a minute when we look at each one individually. And then this would repeat, right? So these thin filaments would extend on the other side of this sort of array of proteins that are sort of holding them, right? And the same thing over here. And then, you know, just to sort of show that it keeps going, these are, you know, the next train in the line of cars in our, or sorry, the next car in the line of cars in our train here, right? So this structure is a sarcomere. Right? That is the contractile unit. And it runs from Z line to Z line. Right? And you can think of it as a Z disc, because remember, think three-dimensionally. This actually have to be kind of, because the myofibrils are circular, cylindrical. This is really a circle in 3 dimensions here. And there would be, you know, fibers that are deeper and fibers that are more shallow, right, going sort of in and out of the paper here, right? So we're looking at a 2-dimensional version of it. So sarcomere goes from the Z disc to Z disc, or Z line to Z line. Again, but you're looking at it 2-dimensionally, it looks like a line. And it is made up of the thick filaments and the thin filaments, the Z protein components that hold the thin filaments in space. We also have a series of proteins. These proteins have to be held in space too. They're not just sort of floating here. And so these guys are held. I'm sure you can see that. Right? And that is the M line. Right? So the M line proteins help stabilize the thick filaments. The Z line proteins help stabilize the thin filaments. And then there is this very essential, and the whole point of this is the sliding filament theory, right? And to have the filaments slide past one another, they have to overlap. So the whole point of all of these proteins that are sort of helping hold things in line and arranging them is to generate this region where this thick filament overlaps with the thin filament. Because again, to have this whole thing shorten, right? When this contracts, separate video, excitation, contraction, coupling. But to have this thing contract, right, we want the Z lines to pull together. To do that, we have to have this filament pull on this filament, sort of pulling itself into this space in here, right? And the same thing true over here. So this guy is pulling itself along. I got think of like a tug of war, right? When you're putting hand over hand, right? This guy's going to pull on the thin filament, pulling it into this space, and the same thing over here. And so these are going to come closer together. And so the whole sarcomere will shorten, which means that the thick filament has to have these sort of hands on it, if you will. They're called myosin heads. And again, I'll draw that a little bit more with more detail in a moment. All right, so I've got these prongs that sort of sit out, the myosin heads, that when stimulated to contract, will grab hold of the thin filament and pull inward there, right? So the whole point of the sarcomere is to generate this, let me put this label in here somewhere, this region right here, right? I'm gonna put my label over here to the side. That is the zone of overlap, right? If you don't have overlap, you cannot get tension. You cannot get tension without overlap because the myosin heads would have nothing to grab onto, right? If we were to stretch this thing and these were no longer overlapping with those, the thick would no overlapping with the thin, right? Thick filaments, thin filaments. What we will find in a moment is that the thick filaments are comprised largely of a protein called myosin, and the thin filaments are comprised of multiple proteins, but the structure of the filament part itself is largely of a protein called actin. Right? So thick filaments are myosin filaments, thin filaments are actin filaments, and again, they have to overlap in this zone to generate tension. Okay, now we said that I want to point out why striated muscle has these stripes, right? We said that striations means stripes. And the stripes come from the fact that you have an A band and an I band, right? So I'm going to put a bracket over the length of the thick filament, right? Just squeeze an A in there. right? So the A band is the length of the thick filament. And I guess because of the density and increased mass of the thick filaments, that appears darker under a microscope. And then you have this region from where the zone of overlap ends, and you go over the Z line into the next sarcomere, right? And it stops right about there, right where the next thick filament starts. And again, okay, now remember, this is the Z line. I'll screw up our labels here. But this space is the I band. Right? And so essentially, if you go along the sarcomeres, you go A band, I band, A band, I band, right? And think dark has the letter A in it, and light has the letter I in it. And so it goes dark, light, dark, light, dark, light, giving it the striped appearance that you see under a microscope. Okay, I think we got most of our functional proteins involved in generating the tension. We have a few proteins that are called molecular rulers. One of them plays more of a role than that. There's one associated with a thin filament called nebulin. Let me just put a little squiggly line here along one of them. This would be found on all of them. Nebulin. And nebulin basically helps the thin filament sort of during formation, like be oriented properly, right? To sort of line up in the way it's supposed to. The other thing, the other protein, and this one's associated with the thick filament, is probably even more important because it's got a functional role in addition to sort of helping align the thick filaments. It's called titin. The newer term for it is connectin. And it is definitively sort of spring-like. And it actually splits at the end. We don't have to worry about that. But what it does is it connects the thick filament, the myosin filament, to the Z line. And so it kind of helps keep it all right. There would be one going this way, right? This sort of spring-like protein. And again, that's we'll call it titin, but it's also got this name. The newer term for it is connectin. Its claim to fame is it's the largest protein in the human genome, enormous protein. And so it's helping the myosin filament, the thick filament, remain aligned here and sort of stay between the thin filaments the way it's supposed to. And in addition, because it's sort of spring-like, when a muscle gets stretched, and again, sarcomere after sarcomere after sarcomere in a line, they all get stretched. When a muscle gets stretched, because of its spring-like nature, it tends to generate recoil and adds to the elasticity of muscle, right? So it's tending to sort of limit passive stretch of the muscle. Okay, now keep in mind, one thing I wanted to point out, right, is that if we were to like cut through this, I'm going to cut through this right here at the zone of overlap, and then I want to turn it so that you can see it end on the ends, right? And so now these filaments will look like little circles, right? Because you're looking at the end of the filaments, right? If we cut it and then turn it so you're looking at the end of it, and again, it would be circular because the myofibrils are circular, and you would have I'll just draw a couple of them. You would have the thick filaments in black, right? And the thin, which are in blue, would surround them. Like so. And it's a very specific number, and I'm not trying to be absolutely correct about everything here, but so each thick filament is surrounded by the thin filaments, and then, as I said, the heads of the myosin, they don't, right? It's not a two-dimensional. They actually go all the way around the myosin filament, so they stick out all the way around to associate with, or potentially associate with, you get the idea here, hopefully, to associate with the thin filaments, right? And again, they will only do that upon stimulation, which is really a calcium release and an action potential in the skeletal muscle fiber, right? But in that way, the thick filaments are surrounded by these thin filaments and can pull on all of them all the way around it to shorten the sarcomere overall. Okay, let's look at the individual filaments. The important ones, the thick filament and the thin filament. So the thick filament is pretty easy. The thick filament is a polymer of myosin. So, right, so myosin is a single molecule protein strand, whereas the thick filament, which is essentially a myosin polymer, is made up of several hundred of these myosin monomers, if you will, components. And so an individual myosin monomer looks a bit like this. It's got this tail-like section, and then it has two heads that sort of stick off of it, and they have special active sites, right? And so we've got this tail, and then we have the heads of the monomer myosin molecule. And so what we do is we take, so again, the heads of these parts that stick out here. The heads are the part that can grab the actin. So the head, the two parts here that I'm pointing to, the two little spots, are the two sort of really important functional regions of the myosin molecule. And that is, they have a binding site for actin. That should make sense, right? And they're an enzyme, right? So one of these sites is an ATPase. In other words, it is the part that in muscle that gobbles up the ATP to generate tension, right? So one of them is an ATPase, and the other one is an actin binding site, right? And so there's a cycling of sort of binding, pulling on the actin, which is that energy was stored by utilizing ATP initially, right, to sort of cock the head in a position so that it's got stored energy in it, and then it releases, and we have to use it, bind ATP again, break the ATP, restore the energy, and so the heads kind of go back and forth, right? So they get cocked with ATP, and then they bind and pull, right? And then they have to release again from the thin filament, right? They get recocked with ATP, they bind, they pull, release, right? Recock with ATP, I think this is longer, right? Bind, pull, release, recock, bind, pull, release, recock, right? And this thin filament just keeps being pulled past. Now, okay, that's what each individual monomer component of the myosin is doing. But keep in mind that the thick filament as a whole is a bazillion, not bazillion, but several hundred of these myosin monomers, right? So there's one, and they sort of overlap, right? And here's another one. Here's another one. You get the idea, hopefully. So this myosin, and here's the thing, right? Here's that, remember that was the M line in the middle that's sort of holding it up. And then the monomers on the other end are sort of facing the other direction with the tail. The tail's always faced towards the center. The analogy I like to use is taking a whole bunch of golf clubs. The golf clubs would have to actually have two heads on them. And put all the handles together towards the middle, and the heads of the golf clubs are all facing outward here, right? And so you get this big polymer molecule, and so on, right? With all the myosin heads sort of sticking out all the way around. And it's right? So here they are, the myosin heads sticking out all the way around, able to bind the thin filaments that should be surrounding it. Okay? And again, each one of these myosin heads has these active sites for binding, and then the energy conversion component, this ATPase enzyme, that uses the ATP to sort of ratchet the head back and forth as it's pulling on the thin filament, right? Okay, so let's look at the thin filament. The thin filament's a little bit more complicated. So the thin filament is made up of Again, lots of monomer molecules called G-actin. And you want to think of those as, the analogy I like to use is pearls on a string, right? And so take a pearl necklace, and grab it end to end, right? Or imagine it, and twist it, right? And so you would have the two strands, right? If you grab this, the necklace, right, and pull it between your fingers and twist it, right? And the two strands will be wrapped around each one another, right? Kind of like a helix, right? Like DNA has a helix structure. And so you end up with these monomers, the blue dots here, sort of twisted around one another. My blue pen's dying. Right? And so that would be one strand. The other strand right, twisted around this one, crossing over. And hopefully you get the idea. Right? So it's sort of like, hopefully that looks a little bit like a helix. Right? And so each one of those is a G-actin molecule. That's sort of the monomer. Well, they're sort of like linked together, if you will, right? Like so. Right? And this one's linked to its. Makes it a little bit more helical looking, right? And so each one of these, though, strand of pearls, if you will, is called F-actin. Well, let me back up. So the G is for globular, like a blob, right? So each one of these is like a blob, like globular protein, right? And then the fact that you string them together to form these strands that wind around each other, each one of the strands is called F-actin, right? So this whole thing, the whole strand equals F-actin, right? And again, one of the strands is F-actin, F for filamentous, right? Like a filament, like a strand. The whole thing together, the two strands wrapped around one another, are the actin filament. Right now, it's not the thin filament yet because we have to add a couple more proteins to it, and then we have the full structure. But so G-actins are the individual monomers that link together to form F-actins, right? F-actins are the strands of G-actins. Twist two F-actins together and you get the actin filament. So that's the whole thing. Again, my labels are going to be a little confusing here because I'm going to add other things to it, right? But just this strand of the F-actins, two F-actins wound around each other, is the actin filament. And then we need the component. So here's the key bit, right? So the myosin head had a binding site on it for actin. Well, those are individually on the G-actins, right? So each one of those G-actins has a little spot sort of facing outward, right? I'm not going to draw them all, right? That would be available to bind. Maybe I'll put them all on here, most of them on here. Right? So that's the myosin binding site. Here's the thing. Myosin and actin stick together like, white on rice. They are glued together, right? When they have the ability to do so. So they want to interact, quote unquote, want to interact. They're highly attracted to one another chemically. There's a binding site, an affinity for binding between the myosin head and the G-actin molecule. So if we want to avoid having contractions all the time, right? You don't want your muscles permanently contracted. We need to inhibit that tendency to form an actin-myosin linkage, right? And that actin-myosin linkage is called a cross bridge. Sometimes people refer to it as the actomyosin complex, right? Actomyosin, the combination of the two. We'll just refer to it as a cross bridge between the thick and the thin filament. Okay, so how do we keep the thin filament from just spontaneously binding to the thick filament component that it wants to, quote unquote, wants to bind to, right? Well, there's a protein that sits in the way of that binding when muscle is not being stimulated. And it's another filamentous protein that's going to sort of sit here right on the surface, and it sort of winds around. It forms these like segments, basically, and it sort of winds around. I'm just sort of drawing it like this, and sits on top of the G-actin binding sites for myosin. And that protein is called tropomyosin. So tropomyosin is it's called steric inhibition. Steric, S-T-E-R-I-C. Just basically means it's in the way, right? So it's in the way. This tropomyosin is in the way of the myosin heads binding to the G-actin. Again, unless we stimulate the muscle. Okay, so how do we regulate tropomyosin? In other words, we have to get it out of the way if we're going to get tension development, right? If we're going to get cross bridges to form. Cross bridges are the basis of muscle tension development, right? You have to have the thick bind and pull on the thin, sliding filament theory. So where's the regulatory protein? Well, that guy is, running out of colors here, here we go, is another protein. It sits right on top of this. I'll put a little binding site on it. It's a little V shape. Right, so each one of the tropomyosins would have this other regulatory protein sitting on it called troponin. Now, troponin's got multiple sort of components that do separate things, but for our purposes, all we really need to know is that it has a calcium binding site. Let's label that. So troponin is the calcium binding protein in striated muscle. So we'll do little dots here as our calcium molecules. All right, so well, let's back up. When a muscle is relaxed, intracellular calcium levels are low because we haven't stimulated its release from the sarcoplasmic reticulum. See the last video, right? And also the excitation contraction coupling video. But so when we stimulate muscle, free calcium levels rise, and calcium is more likely to bind to these sites, right? And so some of these sites, and the higher the calcium levels go, more sites we would sort of bind to, right? More and more calcium, so this is calcium. Right? And that calcium ion binds there and activates the troponin, and the troponin shifts the tropomyosin off of the actin filament, or well, really off of the, not literally off of it, but sort of out of the way, so that the G-actin site for myosin binding is exposed. Right, so all these little black dots were where the myosin could bind, but only if we shove the tropomyosin out of the way. The troponin, upon binding calcium, is what shoves it out of the way. Now all of a sudden you have everything you need to generate tension. The binding site is available here, it's exposed, right? The myosin head, as long as you have ATP as well, as long as you have ATP, the myosin head is just going to start grabbing and pulling and grabbing and pulling and go through its cycle of bind, pull, release to generate shortening of the sarcomere overall. Okay, and again, reiterating what's in the excitation contraction coupling video, the way we get a contraction to go away is reduce intracellular calcium levels. It will diffuse off of the troponin. The troponin will no longer be moving the tropomyosin off of the myosin binding sites. The tropomyosin will sort of shift back into place and sit in the way of binding with the myosin, the adjacent myosin heads. Okay, so we've got our sarcomere structures, right? The accessory proteins that sort of hold things in place, like the Z-line proteins and the M-line proteins, and titin and nebulin that are sort of helping align the thick and thin filaments. And then we looked at the thick versus the thin filaments and their basic structures. and then hinted at some of the components in the excitation-contraction coupling by talking about how calcium regulates the troponin-tropomyosin complex to remove the inhibition of cross-bridge formation, right? And again, cross-bridges are the basis of muscle tension production, and they are, a cross-bridge is the connection of the myosin head to the actin molecule. And remember, as long as you have that exposure because the tropomyosin is out of the way and you have ATP, you will get muscle shortening.